Down at the bottom of page 25, let's go ahead and draw some pictures to help us remember what these terms mean. So I want to go ahead and split each of our boxes with the dotted line, and this is going to represent a semi-permeable membrane. On one side, let's draw in a bunch of dots, and these dots are representing the solute particles. On the other side, we're just going to put a few dots. Now, in this first box, which side is hypertonic to the other? If you said the left, great job. Go ahead and draw an arrow to remind yourself that this side is hypertonic and this side is not. In the middle square, let's go ahead and draw the exact same situation. So a ton of solute molecules on one side and just a few on the other. In this case, which side is hypotonic? The one with less solute. So draw an arrow to remind yourself that this is the side that is hypotonic. And the last situation is isotonic. So we should have the same number of solute molecules, or the same concentration of solute, on both sides. And now both sides are isotonic to each other. Go ahead and turn to your neighbor and talk about these three questions. If you're having a hard time with these types of problems, draw yourself a picture. You can draw a simple diagram with a lot of dots to represent the hypertonic area and just a few dots to represent the hypotonic area. So if we're putting an animal cell in a solution where there are more dissolved substances on the outside, the water is going to rush out of the cell and the cell is going to shrink. If we have more dissolved substances on the inside, water is going to rush into the cell and the cell is going to swell and potentially burst. When there are equal amounts, water is still moving back and forth at the same rate, so we're going to see no net movement of water. So far, everything we've talked about has been passive transport. Let's shift gears and move into active transport. This is at the top of page 26 of your packet. Active transport is the movement of solute against the concentration gradient. So now instead of going from high to low concentration, we're going from low to high. Since we're pumping against the flow, this is going to require some energy in the form of ATP. Here are a couple examples of what active transport might look like. Notice that there's a lower concentration here, a higher concentration here, and we're moving from low to high. This ATP right here is showing you that we are in fact using energy because we're going from low to high, and that's why this is called active transport. Here we can see a specific example of active transport. This is called bulk transport. So you can see there's a ton of molecules on the outside, and we want to bring a bunch of them in at the same time. The cell membrane can pinch off and form a vesicle and bring materials in. We can do the exact opposite and release materials on the outside by fusing that vesicle with the cell membrane. If we're bringing materials in, this is called endocytosis. Endo kind of sounds like enter, so we're bringing particles into the cell. There's two types. There's phagocytosis and pinocytosis. Phagocytosis is kind of like a cell eating because it's going to take in a large food particle or other material, but it's going to be large. That's what defines phagocytosis. You can see that this amoeba is pinching in around that food particle and forming a little food vacuole. Pinocytosis is when we're taking in either small molecules or liquid, and this is kind of like the cell drinking. Exocytosis is instead of bringing stuff into the cell, it's exiting. So exo sounds like exit. Here we can see how this little vesicle is going to fuse with the cell membrane and dump the contents on the outside. This is how the cell is going to get rid of waste. Here's another quick recap. We have passive transport, where we're going from high concentration to low concentration. If those molecules can just move straight across the phospholipid bilayer, it's called diffusion. If they need a channel protein to help them get across, it's called facilitated diffusion. Active transport is going to require energy, and we're going from an area of low concentration to an area of high concentration. Go ahead and talk to your neighbor about what's going on in these 4 animations. Here's your last chunk of review questions. Go ahead and talk to your neighbor about these 4. Active transport is going to be different from passive because we're going from a low concentration to high concentration, and we're using energy to do that. The cell membrane is going to help bring in particles. Large particles are going to come into the cell using phagocytosis, and small or liquid particles can come in using pinocytosis. Go ahead and take a couple minutes, work with your neighbor, and see if you can answer this bottom half of page 26. Pause now, and then we'll check your answers in a second. I actually want to start at the very bottom. So in this case, we're trying to describe the solution that the cell is in. This first one, we have a lot more solute on the inside than the outside. So we can call the outside hypotonic. Remember, hypo means low. In this middle case, we've got an equal solute concentration on the inside and outside. So this is an isotonic environment. And in the last case, we have a lot more solute on the outside than the inside. This is going to be a hypertonic solution. Now, when we place a cell in an isotonic solution like we did here, we are not actually going to change the shape of the cell because we have an equal rate of water moving in and out of the cell. When we place a cell in a hypotonic solution like we did here, water is going to move into the cell, and that's going to cause the cell to swell. When we place a cell in a hypertonic solution like this case, water is going to move out of the cell to help dilute the environment, and that's going to cause our cell to shrink. Now, go ahead and move on to page 27, work with a partner, and play when you're ready for some answers. All right, let's take a look at page 27. In this case, our cell has 95% water on the inside, and the outside is a 5% water solution. We have a higher water concentration on the inside and a lower on the outside. Water is going to move from high to low, so it's going to move out of the cell. Because water is moving out of the cell is going to shrink. Number 2, we have a 95% water solution on the inside, 95% water on the outside. That means water is going to flow equally in both directions. And the cell will stay the same. In our last example, we have 5% water on the inside, 95% water on the outside. Water is going to move from high to low, so water is going to flow into the cell, and the cell will burst. Remember, when we say that water is moving from high to low, we're talking about a higher concentration of water to a lower concentration of water. If we flip the page to page 28, we've got a really great flowchart. And I want to make sure that we're just putting down the important ideas for each category. With passive transport, the 2 main things to remember is that there is no energy required, and that we're going from a high concentration to a low concentration. Active transport is the exact opposite. So this time we will need energy input, and we're moving from low concentration to high concentration. Within passive transport, we have 3 different types. Diffusion is the movement of solute. Facilitated diffusion is the movement of solute through a protein channel, and osmosis is the movement of water. All 3 of these are examples of passive transport, so they're all going to require no energy, and we're always moving from high to low. Active transport, on the other hand, we have endocytosis and exocytosis. So endocytosis is when we enter the cell, Exocytosis is when things are exiting the cell. And both of these are types of bulk transport. So we're moving lots of stuff at once. Within endocytosis, we can have phagocytosis, which means we're bringing in large particles, or pinocytosis, which is small or liquid particles. And remember, the point of all of this, the importance of cellular transport, is to maintain, here's your key word for the day, homeostasis. That's the key to it all.